Thank you. Okay, good. Hi, everyone. My name is Aaron Baker with the University of Wisconsin, and I'll be talking about pathway-regularized matrix factorization. So first, why are we interested in matrix factorization? This is a popular family of techniques in machine learning for finding low-dimensional substructures or signals that might be present in high-dimensional data. So algebraically, this takes the form of the equation written here, and visually, we can think of this through these heat maps below. So the data takes the typical form of a machine learning data matrix, where we have observations along the rows and features along the columns. And we're trying to decompose that matrix into the product of two matrices that are much smaller rank than the input matrix. In particular, I'd like to focus this talk on the sort of W matrix, which is the matrix on the right, which is the one that clusters features together into different latent factors. And I say clusters because you can think of the output of matrix factorization techniques as a clustering. In traditional clustering methods, we might associate a feature to one cluster by giving a binary score in one of the cells in this heat map, and further require that the ones that we're assigning are sort of mutually exclusive across the rows. But matrix factorization might give a more of a soft clustering view, where each feature is allowed partial membership into different latent factors. So for biology, the typical data we might see comes from a variety of different high-throughput experiments. This could come from microarray data, or it could come from experiments that are seeking to measure protein phosphorylation levels sort of across all proteins in the proteome, or a variety of different data formats. And past research has shown that these matrix factorization techniques are quite effective in finding important latent factors. So one example, again, talking about that sort of W matrix that clusters together features or maybe gene measurements together, is a study that found that when analyzing the genotype tissue expression data, or GTEx, that data is readily decomposed into latent factors that are associated with sort of tissue-specific gene expression patterns. And furthermore, the other matrix, which clusters together different samples, the H matrix, that matrix has been found to separate disease and healthy samples from each other. So one problem with matrix factorization techniques, especially non-negative matrix factorization, which we use in this work, is that the methods can be pretty unstable. The reason for this is that the optimization procedure to find the matrix factors typically is an iterative procedure that can only find a locally optimum solution. And because of this, different runs of the algorithm might produce different results. One way to overcome this and also add some biological interpretability to the results is to include some prior information. So some past work has done this, including network-based stratification, which used priors of the form of a protein interaction network. However, one problem with this is that protein interaction networks tend to collapse data from all different types of contexts, and so we lose some of the sort of context-specific information that might be present in our latent factors. Another method, the pathway-level information extractor, or plier, sought to include some of this context-specific information from pathways, but it did it in the form of gene sets. So when looking at pathways, plier looks at which genes are members of that pathway, but it doesn't include any of the interactions between genes that sort of define how that pathway operates. And we thought that those types of interactions between genes are an important piece of information, so we sought to develop an algorithm that included that type of information as well. So here is an overview of pathway-regularized matrix factorization. You can see that we're still doing matrix factorization, just like the slides before, but we've added these arrows that are illustrating that we are seeking to constrain the latent factors that are associated with different features. Again, these could be measurements on gene expression, such that the latent factor patterns are consistent with a pathway. And each latent factor can be sort of consistent with a different pathway. And the way we do that is through these regularization terms that I've kind of put in pseudocode, the manifold regularizer and the ignore penalty. And we'll talk about that on the following slides. So before we can do that, we need to find a way to represent the pathways in a sort of linear algebra framework so that we can use them in this optimization procedure. So the way we do that is we first might download a pathway, such as this influenza A pathway excerpt that I'm showing in the top left, and represent that pathway, which includes all sorts of different signed and directed edges describing the relationships in the pathway, to an unsigned and undirected graph, which is being represented below. And we can use graphs of that form in the form of a Laplacian matrix. And I'm sure many of you are familiar with this, but this matrix has along the diagonal an entry for each node, and the value there is the degree of that node, whereas the off-diagonal entries that are highlighted are negative entries that are corresponding with each edge in that network. So the way that we can use these Laplacian matrices is illustrated on this slide. So the main purpose of our method is to try and find patterns that are consistent with pathways. So this illustration is showing how these latent vectors and this heat map on the left are consistent with pathways. So this is showing two candidate pathways that we could think might be consistent with this latent vector or not. And the way we're measuring this consistency is through what's known in the literature as manifold regularization. And so in the figure on the left, we can see that the latent factor is quite consistent with this pathway because the latent factor assigns high scores to nodes 2, 3, and 4. And these are exactly the nodes that are sort of in their own connected component in this first pathway on the left. But if we look at a second candidate pathway, the one on the right, L2, we see that the main difference between this pathway and the other pathway is that there are these additional edges between nodes 1 and 2 and between nodes 4 and 5. But these edges, if we look back to the latent vector, have the property that the nodes 1 and 2 have very different scores from each other, as do the nodes 4 and 5. And as a result, this association would not be a very good association. And so there's a higher penalty to be paid in our optimization procedure for making this type of association. So one way to sort of pay a low penalty is to simply ignore the pathways altogether. And this is especially problematic because usually the data sets that we're looking at are making measurements genome-wide. So there might be 20,000 genes in our data matrix, but pathways are typically defined on only 200 or 300 nodes. So we need to encourage the method to not cheat. So you can see this type of cheating behavior in this first figure on the left, where the latent factor simply doesn't pay any attention to nodes 2 through 6, which are the pathway nodes. And if we don't do anything to correct this, there's no penalty to be paid. So we add a penalty term for ignoring the pathway, also known as a manifold, which is represented in this second equation on the left side. And you can see that this is quite a high penalty versus the situation on the right, where the method actually does pay attention to the pathway, and it does pay some penalty for violating the smoothness constraints imposed on the graph, but it's not anywhere near as severe as ignoring the pathway altogether. So now we have a way to encourage the method to look for patterns that are associated with these pathways. And here is the full optimization procedure for doing that. This optimization objective function is a smooth function, so it's readily solved by gradient descent type algorithms with an iterative procedure. And actually, we can take advantage of the fact that we're doing an iterative procedure when trying to make these associations between latent factors and pathways. In particular, it might be challenging to find which pathway we should be associating with a given latent factor because there could be many pathways. For instance, in the KEGG database, there might be over 300 pathways, and in Reactome, over 2,200 pathways. So just to facilitate the optimization and recognize the type of penalty for ignoring the pathways, we can kind of score pathways as we iterate by just noting, again, how the latent factor kind of ignores that pathway or not. So we have a sort of filtration process when trying to find this minimum term in the middle, where we just note which pathways seem to be ignoring the latent factor. So that pathway on the right is being filtered out because it has very low scores in the latent factor. So having described our algorithm, we then sought to evaluate the performance of this method on two different data sets. One of them is a simulated data set that's meant to mimic genetic screening data. So in genetic screening experiments, researchers are typically knocking out genes one by one, genome-wide, and seeking to measure the effect of that knockout on some phenotype of interest, maybe it's survival of a cell. So to simulate that, we said that there might be a few pathways responsible for our simulated phenotype. So we sample a few pathways from KEGG. And then from those pathways, we'll sample a few of the pathway members along with some noise and put those members into a binary vector on the left. Then we'll diffuse the scores in that binary vector across a protein interaction network. So we arrived at a smoothed observation vector. That's the vector on the right. And finally, we'll consider that one of many observations that we'll generate through this simulation. And then we finally are able to build up a sort of typical data matrix that fits into the input of our method. So having done that, when we look at the results of our procedure, we can look at one of the latent factors as a binary classifier. And these latent factors have scores on different genes, and we can view those as confidence measures in predicting whether or not those genes are members of the pathway that we used in the simulation or not. And thinking about it in this context, we can then look at typical metrics for measuring classifier performance, such as precision recall curves. And I'm showing the precision recall curves for our method along with traditional NMF, or non-negative matrix factorization. And you can see that PRMF performs better. And in particular, the precision values are very high for the early recall levels. This means that the most confident predictions are accurate. And this is particularly important in biological applications because we might only look at our most confident predictions for follow-up studies in the lab. So another data set that we use to evaluate the performance of our method is from Project Achilles from the Broad Institute. This data set is also a genetic screening set of experiments, and they're looking to define the cancer dependency map. So they're analyzing hundreds of different cancer cell lines and performing CRISPR-based gene knockouts of different genes in those cell lines and looking to see which genes are essential for that cancer cell line to survive in a sort of cell line specific manner. So when we run our method on this data set, we find that one of the optimal latent factors is associated with the MAPK signaling pathway. This is a great sanity check for us because this is a cancer data set, and the MAPK signaling pathway is a well-known cancer pathway. It's involved in controlling apoptosis, cell differentiation, and proliferation. So this is a great, just a sanity check to measure our performance against. And then one of the more interesting aspects of our model is that we can start to analyze other genes in the latent factor that are not members of the pathway, but might interact with that pathway in a global protein interaction network. And so we might be able to make some novel insights into ways to extend the pathway by looking at high-scoring genes in that latent factor that's associated with the MAPK signaling pathway. So just to highlight one of these genes, the TED1 gene, we actually heard about this family of proteins earlier in the keynote today, but this protein is a T-domain binding protein, and it's also involved in what you might expect a sort of cancer signaling pathway controlling cell size and apoptosis as well. So just to conclude, we find that this algorithm, Pathway-Regularized Matrix Factorization, is able to guide latent factors to be more biologically meaningful. And this is true for some simulated data as well as the Ivana cancer dependency data set. If you'd like to use these methods yourself, they're available on GitHub. And if you'd like to learn more, come by my poster later this evening. I'd like to thank the members of the Gitter Lab for providing helpful feedback on how to express some of these ideas, as well as funding that made this trip possible. Thank you.
We have time for questions.
Thanks. That was really interesting. Do you have any thoughts about a way forward for incorporating more detailed mechanistic information in these prior networks? So, for example, direction or sign in these types of approaches?
Not at this time. It's actually quite challenging because the matrix factorization we chose for non-negative matrix factorization makes it difficult to incorporate sign. So we'll have to investigate that more thoroughly in the future. Yeah.
One comment that is very related to the previous comment is the fact that within pathways you sometimes have nodes that basically their downregulation indicates activation of the pathway. At least from what I gathered from the formalization you presented, right? Those would actually be penalized under the. Right, so that's question one. And the second question is I think one of the limitations of some of these techniques is our difficulty to scale them. Right, there are some really clever algorithms like the block pivot method, right, for doing non-negative matrix factorization that don't really like it's hard to formalize them using these regularizers, right? So do you have any thoughts about this? Like how well does this scale A, right? And then what do you do about parts of the pathways that are basically reverse in sign?
Right. Yeah, I think I don't have a good answer about the sign problem. It's something we're thinking about. I think that we'll need to look at a regularizer that's separate from the manifold regularization technique to be able to incorporate those ideas, or maybe separate the sort of positive and negative edges into two separate networks or something like that. But it's something we're concerned about as well. And then for the scalability, I think by using some of the maybe heuristics I showed about doing some of the filtering as we iterate in the procedure, the performance runtime is actually pretty good. And all the data I've shown here was generated just from running the algorithm on my own laptop, so it doesn't take too long, I think. So.
Okay. Oh.
I think you were here
Okay,
first.
so quickly, so you said there was the problem of the dependence on initial conditions, but that can be addressed with Bayesian probabilistic matrix factorization. So did you compare?
Not yet. This is kind of early work, so our only benchmarks so far have been sort of traditional NMF. The kind of initialization technique for sort of non-Bayesian NMF is something we looked at. So at least in that context, the initialization, yeah, the performance wasn't as good. So.
Okay, thanks.
Okay, it really is not a question, but this is obviously relatively early work. But I'd encourage kind of calculating anything like this, and it's just a general comment for NetBio. You know, several metrics. One is what happens if you somehow kind of use a random network as a background. Okay, and probably kind of do it 1,000 times and calculate the background probability. The other thing is what happens if you use a complete network. Okay, and what happens if you use different networks? Because it is so easy to fool yourself, with that type of thing. And we probably have somehow, enforced these standards. I mean, this goes back to 10, 20 years. That is part one. The part two is, semantics matters. So you first start describing GTEX, right, which is gene expression, and then there's the project Achilles, and then all up to so the semantics of using a background pathway for synthetic lethality is very different than RNA-seq or gene expression, right? So I think it's really important to kind of formulate right from the beginning what the semantics of the pathways is and how you're mapping it and what the real biological hypothesis underlying it.
Yeah, thanks. Those are good comments, and yeah, it's great to hear feedback like this because yeah, it is early work, and we'd like to incorporate the ideas from the community as well. Thank you.
Thanks, Aaron. All right, now for something a little different.